The third kingdom that we'll study is kingdom fungi. Take a moist piece of bread or bhakri. Moist is slightly wet. So you have to take a slightly wet piece of bread or bhakri and keep it in a container, that is in a box, with the lid on for 2 to 3 days. For those 2 to 3 days, you don't have to open the lid. After 2 to 3 days, a fine cotton thread like tuft is found growing on the surface of the bread. So after 2 to 3 days, when you open the lid, on the surface of the bread, you will find a fine cotton thread like substance. So you have to take that and prepare a temporary mount of it, of those few threads from this culture, and observe it under the microscope. So what are the characteristics of kingdom fungi? These are non-green eukaryotic heterotrophic organisms. So they are non-green, they are eukaryotic, is that their structure of the cell is organized, and heterotrophic is that they cannot make the food themselves and they acquire their food from outside. Most of them are saprotrophs, that is, they acquire their food from other dead organisms such as plants and animals. They feed upon decaying, that is, dying organic matter. Their cell wall is made of tough and complex sugar called chitin. So cell wall is a layer present around the cell membrane, and it is made of tough, that is strong and complex sugar, and the name of this sugar is chitin. Some fungi are thread-like and many nuclei are present in cytoplasm. So when you see the fungi under the microscope, they appear thread-like, and inside one cell there are more than one nuclei. Examples of kingdom fungi are baker's yeast. This is what bakers use to make bread. Aspergillus, this is a fungus present on corn. Penicillium and mushrooms. As you can see here, this is an example of a mushroom, and mushroom is an example of kingdom fungi. Though many systems of classification are introduced after the Whittakers, his five kingdom system is widely accepted. So after Whittaker, many other scientists tried to organize all the different organisms into different subgroups, but Whittaker's classification is the one which is widely used. Classification of microbes. Among the living organisms, microorganisms are largest in number. Hence, they are classified as follows. So, among all the living organisms like animals, birds, fish, plants, microorganisms are the one that are largest in number. Hence, they are classified as follows. So, under microorganisms, there are prokaryotes and there are eukaryotes. Prokaryotes have bacteria, and under eukaryotes, we have protista, fungi, and algae. With reference to size of microbes, remember 1 meter is equal to 10 raised to 6 micrometer, and 1 meter is also equal to 10 raised to 9 nanometer. So how we have meter, centimeter, millimeter, there is also micrometer and nanometer. So when you compare these with 1 meter is equal to 10 raised to 6 micrometer. That is 1 followed by 6 zeros. 1 meter is equal to 10 raised to 6 micrometer, and 1 meter is equal to 10 raised to 9 nanometer. That is 1 followed by 9 zeros. So now we will see the different types of microorganisms. Bacteria. So the size of bacteria are usually from 1 micrometer to 10 micrometer. These are unicellular, that is, they just have one cell. They are independent or parasitic organisms. So the way that they get their nutrition is either they are independent, that is, they can get their nutrition by themselves, or parasitic, that is, they get their nutrition by taking it from other organisms. Sometimes many bacteria together form colonies. So when each bacteria divides and divides, they make many cells, and these cells are independent bacteria that together form colonies. Bacterial cell is prokaryotic with a cell wall. So they are prokaryotic, that is, their cell have organelles which are not organized, and these have cell wall but distinct nucleus or cell organelles are absent. That's what makes it a prokaryotic organism. They reproduce by simple binary fission. So what is binary fission? Binary fission is when one cell, let's take this example of this bacillus, this will divide into two, and that will form two different types of organelles, and that will form two different bacteria. In favorable conditions, bacteria grow vigorously and can double their number in every 20 minutes. So if the conditions are favorable, that is, if they have the required nutrients and the temperature and everything is right for the bacteria to multiply, they will keep on multiplying vigorously, that is multiple times, and double their number every 20 minutes. So here are examples of different types of bacteria, and these are classified based on their shape. So coccus is when the bacteria are round shaped. Bacillus is when they are a rod shaped. And coccobacillus is when they are neither coccus nor bacillus. So they are neither round nor they are like a rod. So they are in between. Vibrio is comma shaped. This is when they are like a rod but they are curved. So they form a comma shape. Spirillum is when they are spiraled. So the next type of microorganism is protozoa. The size of protozoa is approximately 200 micrometer. So do you remember the size of bacteria? They were between 1 to 10 micrometer. So since protozoa is around 200 micrometer, protozoa are relatively larger than bacteria. Protozoans are found in soil, fresh water and seawater. Some are found in the body of other organisms and are pathogenic. So what does this mean? So protozoa, when they need their nutrition, they can be present within other organisms such as humans, birds and animals, and are pathogenic. Means if they are present in the other organisms, they will produce diseases in the other organisms. These are unicellular organisms with eukaryotic cell. Means they have a single cell and the organelles within these organisms are organized. There is great variation in cell structure, organs of locomotion and modes of nutrition among protozoans. So protozoans is a very big class with many organisms present in it. So all of them have different types of cell structure and they have different organs of locomotion. Some use cilia for motility and some use flagella. And modes of nutrition among protozoans. That is, different protozoans acquire their nutrition in different ways. These organisms reproduce by simple cell division. That is, one cell will divide and it will form two different types of cells. Examples of protozoa are amoeba, paramecium. These are free living in dirty water. That is, if you find any dirty water and you look at it under the microscope, you will definitely find many amoeba and paramecium living in it. Also, there is entamoeba histolytica. This causes amoebiasis. So, this protozoa, if it is present in us, it will cause a disease called amoebiasis. And in this disease, you will get symptoms such as diarrhea. There is also Plasmodium vivax, which causes malaria. Malaria is transmitted by mosquitoes, and if there is this protozoa within those mosquitoes, they will infect us and cause a disease called malaria. Also, euglena is another example of protozoa, and these are autotrophic. That is, they can produce their nutrition by themselves. Next, we will talk about fungi. The size of fungi are approximately 10 micrometer to 100 micrometer. So, they are relatively larger than bacteria but smaller than protozoa. These are found on decaying organic matter and dead bodies of plants and animals. Therefore, fungi was an example of saprophytic organism because they acquire their nutrition by growing on decaying organic matter and dead bodies of other plants and animals. These are eukaryotic organisms. Some are unicellular and others are visible with naked eyes. So, these are eukaryotic organisms. That is, their cell is structured and the organelles within these cells are organized. Some are unicellular. That is, they are single cell. And others are visible with naked eyes. That is, when you see it with your naked eyes, they can be present on substances like bread and decaying food. They are saprophytic. That is, they acquire their nutrition from decaying organic matter. They reproduce sexually and asexually by cell division or by budding. So, fungi reproduce sexually with the help of spores and asexually by cell division or budding. Cell division and budding is when one cell divides and forms two daughter cells. Examples of fungi are baker's yeast. This is used by bakers to make bread, candida, and mushroom. The next microorganism that we'll talk about is algae. Their size is approximately 10 micrometer to 100 micrometer. Therefore, algae are similar in size to fungi. These are aquatic, that is, they grow in water. They are eukaryotic, unicellular, that is, they have just one cell, and autotrophic organisms. So, autotrophic is that they make their foods by themselves. They use photosynthesis. So, photosynthesis is carried out with the help of chloroplast present in the cell. So, all of the algae have something called as chloroplast, and they use this chloroplast to do photosynthesis. And by photosynthesis, they make their own food. Examples of algae are chlorella and chlamydomonas. Very few species of algae are unicellular. Most of them are multicellular and visible with the naked eyes. So, there are very few species which are just one cell. They are unicellular. Many of them are multicellular, that is, they have many cells, and because they have many cells, we can easily see them with our eyes and we don't need a microscope. So next we will talk about viruses. Their size is generally approximately 10 nanometers to 100 nanometers. Up till now, whatever we studied like bacteria, fungi, algae, they were all in micrometers. But viruses are in nanometers. Therefore, they are very, very small. And sometimes they are difficult to see under even certain microscopes. Generally, viruses are not considered as living organisms, or they are said to be organisms at the edge of living and non-living. So, because viruses are so small, sometimes they do not have all the cell organelles that other cells have. Therefore, they are said to be at the edge of living or non-living. They are studied under microbiology. So, microbiology is a subject that studies micro, that is very small, and biology, that is cells. So, they study very small cells like viruses. Viruses are extremely minute, that is very small. That is, they are 10 to 100 times smaller than bacteria and can be seen only with electron microscopes. So, since they are so small, viruses can only be seen under certain type of microscope, which is called electron microscope. This microscope is very powerful and it is only used to see very small things such as viruses. They are found in the form of independent particles, that is, they are found separately and not in clusters. Virus is a long molecule of DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid, covered by a protein coat. So, therefore, they were called as at the edge of living or non-living. It is because they did not have the same organelles that other cells had. They just have a DNA or RNA, which is covered by a protein coat. Viruses survive only in living plant or animal cells. Because they do not have other organelles, they have to survive by taking nutrition and other substances from other plant or animal cells. So, they infect the other animal cells or plant cells and produce their own proteins with the help of host cell, that is, the plant or animal cell in which they are, and create numerous replica. So, suppose this is a plant or animal cell, viruses infect it and start multiplying using the host proteins. So, once they multiply, they form numerous replica which are of the same viruses. Then, they destroy the host cell and become free. So, in order for these viruses to come out of the cell, they have to destroy the cell membrane of the animal cell. So, they destroy this animal cell and come out and become free as other viruses. These free viruses again infect new cells. So, these individual viruses can each infect other cells. Viruses cause many diseases to plants and animals. So, because they break their cell membrane and because they destroy the animal cells, they can cause diseases in plants and animals.